Hi everyone, I am Estella Murray B. Mendares. So, have you heard the disease called sudden oak death? Well, I'm sure some of you have heard it but do not familiarize with this disease. Well, this disease causes significant ecological and economic damages. It poses a major threat to forest ecosystems, biodiversity, and the nursery industry. So what actually causes this disease? Well, for this video presentation, we will be talking about the Phytophthora ramorum, which is the pathogen that causes the sudden oak death. The picture on the left, or the figure 2, is the symptoms which is caused by the Phytophthora ramorum, while the picture on the right, or the figure 3, is the microscopic images show different life stages and structures of an oomycetes, which is a group of fungus-like microorganisms and come from the genus Phytophthora. So what is Phytophthora ramorum? It is a fungal-like organism or water mold that behaves like fungi but actually belongs to a different group known as oomycetes. It is an invasive plant pathogen that causes several plant diseases such as sudden oak death, ramorum blight, and Phytophthora canker. This organism thrives in moist environments, spreading through rain splash, wind-blown rain, and contaminated soil or plant materials. Based on its classification, Phytophthora ramorum belongs to the kingdom Coromista or Straminopila, phylum Oomycota, class Oomycetes, order Pyronosporales, family Pyronosporaceae, genus Phytophthora, and species Ramorum. This taxonomy highlights its close relation to other destructive water molds that affect crops and trees globally. Although Phytophthora species look similar to fungi because of their hyphal growth and feeding patterns, they are quite different. Unlike true fungi, they are diploid for most of their life cycle and produce swimming spores called zoospores. Taxonomically, Phytophthora belongs to the kingdom Straminopila, a group that also includes diatoms and brown algae, rather than the fungi. Genetic studies of Phytophthora ramorum show the presence of plant-like genes, suggesting that their ancient ancestors may have been photosynthetic organisms. So based from the picture that can be seen on the screen, within the Phytophthora genus, Phytophthora ramorum belongs to clade 8C, together with Phytophthora lateralis, Phytophthora hibernalis, and Phytophthora foliorum. These species are genetically distinct, and their exact origins remain unclear. Based on molecular studies, Phytophthora lateralis is considered the closest known relative of Phytophthora ramorum, although its precise evolutionary relationship with Phytophthora hibernalis and Phytophthora foliorum are still uncertain. The life cycle of Phytophthora ramorum begins with the formation of sporangia, which are structures that release zoospores, or the tiny motile spores that swim in water to find and infect new plant tissues. These zoospores insist, germinate, and penetrate the host plant, leading to infection. Although direct germination from sporangia is uncertain, it occurs in related Phytophthora species. Once infection occurs, the pathogen produces chlamydospores which serve as resting spores that allow it to survive adverse conditions such as dry or cold weather. These chlamydospores can overwinter in infected leaves, soil, or plant debris, ensuring the pathogen's persistence in the environment. Dispersal occurs through wind, water, and soil movement, as well as contaminated tools, footwear, or nursery materials. Under favorable conditions, sporangia are produced again, restarting the infection cycle. Sexual reproduction may occur when two meetings, the A1 and A2, come into contact, forming oospores through the fusion of mycelia. However, in Phytophthora ramorum, viable oospores are rarely produced under natural conditions.
Phytophthora ramorum causes two major diseases depending on the host. In forest trees such as tan oak and coast live oak, it results in sudden oak death. The key symptoms include bleeding trunk cankers, inner bark necrosis, and progressive canopy dieback, which eventually leads to tree mortality. In contrast, on understory and ornamental host, the pathogen causes ramulum bite. Here, we typically observe water-soaked leaf lesions, brown to black necrotic blotches, and shoot dieback, which can reduce plant vigor. An important point is that some plants remain asymptomatic, but they can still carry and spread the pathogen, making disease management more challenging. Phytophthora ramarum has a very broad host range, infecting more than 109 plant species across natural and cultivated ecosystems. Hosts like tan oak and oak species are highly susceptible, often developing lethal trunk infections. Meanwhile, foliar hosts such as rhododendron, camellia, and California bay laurel usually show milder symptoms, but they can produce large quantities of spores, making them primary drivers of pathogen spread. Because susceptibility varies wildly, some plants serve as reservoir hosts, silently contributing to disease outbreaks even when symptoms are minimal or absent. Microscopically, Phytophthora ramorum is characterized by specialized structures that support reproduction and survival. First, it produces elongated to ellipsoid sporangia, which are semi-papillate and can form individually or in clusters. These are responsible for asexual reproduction and release infected zoospores. Second, the pathogen forms large globose chlamydospores with thick walls. These serve as long-term survival structures, allowing the pathogen to persist in soil, water, and plant debris even without a host. Although sexual reproduction is rare, Phytophthora ramorum can produce oospores. These are round thick-walled structures that form when compatible mating types interact, providing additional durability under harsh conditions. Together, these structures explain why pathogen is persistent, highly infectious, and difficult to eradicate.
Phytophthora ramorum is a plant pathogen that was first discovered in 1990s, both in Europe and North America. It was initially found in nurseries, particularly in Germany and the Netherlands, where it infected ornamental plants such as rhododendrons. Later on, it was identified in forests in California and Oregon, causing a serious disease known as sudden oak death. Over time, this pathogen has spread to other regions, including parts of Europe and the United Kingdom, and also Canada. Its spread happens mainly through infected plant materials, contaminated soil, and water. Because of this, it has become a global concern for both forest health and the plant nursery industry. This pathogen prefers cool, moist, and shaded environments, which are commonly found in coastal forests and nursery areas. These conditions make it easier for the spores to survive and spread, especially during the rainy season when water movement helps carry the disease from one plant to another. So, human activities such as transport of infected nursery plants and soil also play a major role in its spread. As a result, many infected areas experience tree deaths, forest decline, and loss of biodiversity. It's very important to understand the occurrence of Phytophthora ramorum because it helps scientists and farmers monitor, detect, and manage the disease more effectively, protecting both natural ecosystems and agricultural crops. Phytophthora ramorum is a destructive plant pathogen that plays a significant ecological role by impacting forest health, biodiversity, soil quality, and wildfire risk. Phytophthora ramorum causes a widespread tree mortality, especially in oaks and tan oaks, which leads to significant forest habitat loss and altered ecosystem structure by removing the key tree species. It also reduces biodiversity by killing host plants and changing the forest composition, which disrupts the food webs and negatively affect the wildlife habitats dependent on those plants. The pathogen contributes to increased wildlife risk due to the accumulation of dead woody debris, which acts as an additional fuel in affected forests.
It also impairs soil health by reducing the biomass of beneficial mycorrhizal fungi, which results in decreased nutrient uptake including the phosphorus and micronutrients, which is essential for plant growth. Phytophthora ramorum acts as an invasive pathogen with high adaptability and a rapid spread, threatening native plants and nursery stocks worldwide, making the ecosystem management more challenging. Effective integrated control and management strategies are essential to reduce the spread and impact of Phytophthora ramorum in forest and nursery ecosystems. First is cultural practices and biosecurity. It implements proper cultural practices and strict biosecurity measures, help preventing the introduction and spread of Phytophthora ramorum in vulnerable areas. Second is chemical control. The use of fungicides can reduce the infection rates and protecting the high-value plants when applied properly and timely. Third is the monitoring and early detection. Regular monitoring and early detection enable the rapid response to new outbreaks, limiting further spread. Fourth is sanitation and host removal. Removing the infected plants and debris reduces the source of inoculum and help contain the diseases. The last is regulatory and quarantine measures. Enforcement of regulation quarantine protocols restricts the movement of infected plant material to prevent geographic spread.